installment of The Last Word, an AALAS podcast. I'm Jennifer Smith, your host, and I'm the Director of Education and Scientific Affairs at AALAS. Today, I'm joined by Dr. Claire Hankenson, who happens to be a trusted colleague, mentor, and a very good friend. Claire is the Associate Vice Provost of Research and the Executive Director of University Laboratory Animal Resources at the University of Pennsylvania. She's also the professor of pathobiology, a member of the American College of Laboratory Animal Medicine, and extremely active and respected in the laboratory animal science community, serving AACLAM, AALAS, as well as AALAC. As an accomplished researcher, Claire's area of interest focuses on rodent models of human disease and refinements in laboratory animal veterinary practice. I am so thrilled to welcome Claire to the podcast today and look forward to hearing all about her recent paper review titled The Elephant in the Room, Recognition and Documentation of Personnel Practices that Confound Reproducibility. So hello, Claire. Oh my gosh. I am so thankful that you are joining us today. And you and I go back a long, long way, sister. I think, if memory serves me correct, we probably connected about 20 plus years ago studying for boards. And I will spare our listeners all of the funny stories about how Claire was very prepared and I was ill-prepared. But nonetheless, we're here today. So anyway, for those of you who might not know Claire, like if you've been living under a rock or something, Claire, can you tell the audience just a little bit about yourself, your background, and you can spare any personal stories about me too.
Well, it is a joy to be here, Jenny. Thank you so much for having me. Congratulations to you for your new role at AALAS as Director of Education and Scientific Affairs. There are so many of us excited to have someone with your expertise and interest in that job. So I'm thrilled to be here. This is my first podcast, Jenny, and I am just so honored because I get to do it with you.
Yay.
So I know you'll be gentle with me.
Of course.
So yeah, I mean, it's when you have a good friend that you can't even remember exactly when you met. I think that's a testament to just, honoring that friendship. Yeah, we joke, we know too much about each other to not be friends at this point.
That's true. That's true.
It is true. It is true. So yeah, anyone is welcome to try to hit us up together at a meeting, and we can maybe share stories. But for today, we'll stick with the professional aspects, of course. I am a laboratory animal veterinarian by training. I went to Purdue University, go Boilers, and I didn't really know too much about animal research until I had to get a summer job when I was a vet student. And then I worked at a dairy and I did a lot of cow vaccine research, interestingly, which goes way back in the time capsule. But I was so interested in research and then still so interested in clinical medicine. And I felt so lucky that I was able to be directed by a small group of very precious mentors in veterinary medicine toward this specialty. And I have been in academic research animal medicine now since the 90s. It's been fantastic. What a wonderful career I've had. I feel very, very lucky and privileged.
Wow. Well, thanks for all that. Thank you for your kind words as well. And we are lucky that you are with us and joining our profession as well as being with us on the podcast. So thanks again for that background. I always think our listeners probably want to know a little bit more about the people that write these amazing papers. And so I really love to ask that question. And I've learned so much about our colleagues. So it's been really fun for me as well. All right. Well, we're pretty excited to delve into this paper. This paper addresses some really crucial aspects of research, and I think this is going to be a must read for scientists as well as, of course, for us laboratory animal scientific people, and for our program. So thanks again. So I'm just going to jump right in here. Your paper starts off with a mouthful, but I would love for you to introduce us to the NIH's Advisory Committee to the Director Working Group, therefore known as ACDWG, not ACDC, as I keep saying in my head to myself as I was prepping for this call. So ACDWG. On enhancing rigor, transparency, and translatability, in animal research. And there was 5 essential themes that you talked about, and it would be really great if you could kind of give us a high-level view of those to anybody who's not as familiar.
Sure, I'd be happy to do that. So just for a little bit of history, because it's important to recognize all of the incredible work that has gone on by so many stakeholders and professionals that we know and many that we don't. But I'll take it right back to where this working group originated. So it really began in 2016, if you can believe that. There was a 21st Century Cures Act that was signed into law right at the end of President Obama's term. And it required in that 21st Century Cures Act that the NIH director, who at the time was Francis Collins, was to convene a working group under the advisory committee to the director, or the ACD. And the goal was to develop policies to enhance the rigor and reproducibility of NIH-funded scientific research. So it was really kind of a call out from the public to the NIH to just do the best job they could with supporting funded scientific research in this country. And so as a result of that effort from the 21st Century Cures Act, Dr. Collins then in 2019, which is when I got involved, organized this working group on enhancing rigor, transparency, and translatability. And it was actually quite a thrill. I got just a cold email from Francis Collins, who's super famous. And I was like, wow, what is this, like spam? Is this for real? But it was real.
I mean, that happens to me every week, pretty much. So.
It has never happened to me before or since, so I'll just be honest. But it was very, very cool. And he was inviting my participation as a laboratory animal veterinary professional to work on this committee, basically, to address at a high level how the NIH might improve the value, rigor, and reproducibility of animal studies. And I know there's a ton of alphabet soup, as you said. It's like ACD, ACDC, all the things, right? So I'll just give a few additional acronyms to the listeners just to help understand where we've come from, because again, there's a lot of different stakeholders who have participated, and I do mention them in the paper as well. So one of them is the Institute for Laboratory Animal Research, which has now actually been sort of rolled into a new board on animal health sciences, conservation, and research, or BASCR is, I think, how they're pronouncing it. There's also the National Academies of Science, Engineering, and Medicine, or NASEM, the NC3Rs, which I'm sure many of our listeners are familiar with, a phenomenal national center for the replacement, refinement, and reduction of animals in research. And I would be remiss if I didn't also mention the excellent work that has come in this area of reproducibility by FASIB, Federation of American Societies for Experimental Biology. So all of these groups kind of came together, were a lot of the background that we looked at as a committee to learn more about what we could do to suggest to the NIH for improving animal research. And you did mention these 5 essential themes, Jenny, and I think for interest's sake, the most important of these was number 4 for me, which was the documentation of methods and results reporting. And that's what I dig into a little bit more in the paper, specifically thinking about what we report in the animal research realm.
Fantastic. And of course, all joking aside, I think that's awesome that you got an email from Francis Collins. I'm, green over here with jealousy, but I'm more thankful too that they were smart enough to find a lab animal veterinarian to engage in this really important topic. So
I realize, like, anyone listening is like, wow, what a nerd. Like, that's what she's excited about getting an email about. But yes, I'm a fully vested nerd for sure, and very proud. Yep.
That's all right. We'll take it. So, and thanks for all of that alphabet soup. Best way to say it. There's just a lot there, but I appreciate you giving credit to those other folks that had a hand in this. And I know there's a lot of work that has gone on pre-2016 and certainly post, so I appreciate that as well. So as I was reading the ACDC, WG, all joking aside, I thought that their definition of extrinsic factors was quite interesting, and I know you delved into that in the paper, and I wondered if you could kind of discuss that with us now.
Absolutely. So this was kind of one of my subcommittee assignments. I got to work with a couple of really fantastic colleagues in that realm. We were out of a larger committee of maybe like 20 or so, a mix of scientists and journalists, a couple of veterinarians, statisticians. It was really kind of a cool group of people, many I'd never met before. But I worked specifically with Eric Prager and Brian Berridge to think a bit more about extrinsic factors. And we got to define this to some extent based on work that had been done previously. So an extrinsic factor for our listeners is probably best boiled down to a factor that has a direct impact on the experience of the research animal during the course of the experiments or in its phases of living in an animal facility. And so this can be how it's housed, husbandry, handling, feed, water, bedding, enrichment, cage type, light cycles, really anything that the animal experiences in that capacity.
Wonderful. So, and I know we're gonna move further through this paper and sort of talk about this elephant in the room, which I love the description there. And I just wondered, you've kind of listed some of the more traditionally known extrinsic factors, and you discuss a few more granular examples in your paper. And I wondered if you could maybe talk a little bit about those.
Yeah, so it's, where this kind of started coming together for me, Jenny, was when I was looking at the fact of all of these different influences, really, that an animal has to, we as people have to help the animals with. It's like there's a person who's there changing cages and a person who's handling the animals, moving them between cages, and a person who's making sure the light cycles work and adding enrichment and food and water. So for me, I wanted to highlight that we have a lot of things that just are in the environment of the animals that we work with. And I also want to be clear too, this isn't just for mice. I mean, we could think about this being for fish or rabbits or monkeys too, right?
Yeah.
Yeah, and so there's a little bit of a nuance for every single type of species that we have, because these feed, water, caging, enrichment devices will all differ, I think, depending on species as well. But I just started thinking about the variety of human beings that have jobs that interface with our animals in the research environment, and we can't control for their behaviors at all, right? We're doing everything we can to control for the animal, but we can't control for the humans so much. So I started thinking about that and just kind of how impossible it is for animal care organizations to accomplish exact reproducibility. And that kind of became the genesis for this thought piece.
Yeah, I love that. That's a great summary. Thank you. Something else I really loved from your paper was this quote. And I was thinking to myself, if I were still directing a program today, I would probably put this on my door so every PI could read it. I'm just going to read it. With awareness of the influences of the animal environment in research settings, one can better appreciate the challenges of capturing and documenting with precision the details of the myriad extrinsic factors. I love it. It's such a great summary. So from all the work that you put into this paper, which I know is extensive, it seems to me that the word awareness is really one of our biggest hurdles. Would you agree?
Yeah, I think so. And mainly it's a difficult word to define, I guess. The way that I intended that was just an awareness of all of these other potential experimental variables. So I think if I was just going to pick out scientist X, for example, and they're focused on diabetes, and they're looking at some manipulation, for example, whether it's a strain background or a transgenic or a gene knockout, they maybe aren't thinking that there could be an influence of blood glucose by the fact that that animal might have fecal pellet ingestion of its own for its own vitamin enhancement, for example, if it's a rodent per se. They may not be thinking about the fact that the metabolism of that animal could be really impacted by the temperature in the room. They may not be considering that if another scientist or research staff member comes in after hours and flips on the red lights and turns on the hood in the room, that may add some stress that could impact metabolism, potentially driving up blood sugar. So there's all of these other things that happen outside of the day-to-day focus on the experiment that's described in a funded grant. And so that awareness, to use that word again, I was hoping we could have more conversations with our scientific partners about how to track all of these potential variables so that they would just have an ability to document them and look back on them as part of the data set.
I completely agree. And like I said, I love that quote, and I love your examples and, just kind of your definition of awareness there. Cause I feel like that's something that's kind of missing in that connection as we help to serve the scientific community. So, like your title, your paper also offers some really good examples of times when studies tried to attempt to control some of these animal and personnel variabilities. And you did a great job summarizing them in the paper. And I just wondered if you could kind of speak to those now.
Sure. Gosh, I think as I thought about this, Jenny, as I was writing this, it becomes a little overwhelming. There are so many different attempts of variables we are trying to control. And it takes a lot of effort, and it's almost impossible to do. So that's really the message that I wanted to get across. But there are many publications, not only within the research animal field, the AALAS journals, as well as ILAR journal, the former ILAR journal, that talk about influences like the light cycles. So there are times where we have, very commonly defer to a 12-12 light cycle, 12 hours of lightness to 12 hours of darkness, for those who maybe haven't heard that term before. But there's also manipulations that can happen where we maybe do a 14-10 light cycle to enhance breeding for particular rodents or potentially for non-human primates. So lighting is just one thing that can differ. Social and behavioral factors have to be addressed. So we've had a lot of push since the last edition of the guide came out to make sure that we socially house social species, and that influences their behavior, whether they're housed with one or more similar animals, and how we're learning more and more about behavior across many, many species. One topic that actually had a lot of heated debate, surprisingly, in that working group was just around the microbiome, or an animal's microbiota, because it's such an important thing now for many, many studies, how the microbiome, or the collection of bacteria and viruses and agents within our gastrointestinal tract, or an animal's gastrointestinal tract, can influence, you maybe heard like the brain-gut axis, for example. Even behaviors can be influenced by the microbiome. But other scientists on the working group had no interest whatsoever in the microbiome. They could have cared less what you were feeding and watering your animals with, because they were looking at something completely different.
Yeah,
but it's really interesting. Some of these things people just didn't even think mattered, but they're incredibly important for other scientific outcomes. So things like drinking water, which I've studied a little bit of influence on drinking water because of how we purify it, because of the microbial and chemical contaminants that it might have. Feed types are really important, again, across species. Bedding and enrichment, I've mentioned these as well. They can influence how animals interact with these substrates in their caging environments. I'll just stop there. There are so many, but the hope was from the working group that all of these would be recognized, documented, and disclosed in protocols and publications to just help other scientists try to repeat these same efforts successfully.
Oh, hear. So I'm sitting here shaking my head like, we're in the same room saying, yes, preach it. Yeah. Yes, absolutely. That's why it's just so exciting to be able to really unpack this paper and share it with our listeners, because it's so important. So I think now that we've clearly identified the elephant in the room, I love that you delve into the importance of animal care and the importance of our animal care technicians, the importance of all of us doing animal handling. And you summarize some really important findings, some sort of hard to read, when you think about, potential pathological effects and things that could happen. And could you kind of summarize that for our listeners as well?
Yeah, definitely. And I did intentionally highlight the colleagues we have that work as animal care technicians, how important that role is. I mean, one of their primary things to do is check animals every single day and make sure that they're in, secure and clean environments. They do the cage changing, et cetera. But I wanted to also make sure I highlighted in the podcast and to the listeners that when I was talking about animal personnel, it really could be anyone that comes in contact with an animal. So that I wasn't trying to, distinguish in any way between a research staff member who may be also manipulating their animals from an animal care technician, because it's the most common research interaction we have between the animal handler, let's call them that, and animals, really at the times of cage change, experimental manipulation, and then as veterinarians when we're doing physical examinations on the animals too. So one of the things that had come up and had really made me start thinking about this, and I need to give credit to some colleagues here at Penn, there was a study, sort of a retrospective study done by a few pathologists at our vet school here, where they did a post-mortem assessment of 1,000 mice used in research. And they divided those animals into about 864 that had been what they called heavily manipulated, moved, had injections done or blood collections, et cetera. And then 136 that had only been handled for routine husbandry procedures, cage changing, et cetera. And what was found was a bit surprising. And as you mentioned, a little unsettling, but it was incredibly valuable information because this is the first paper of its kind that I've seen to think about what we're seeing as kind of post-mortem lesions, if you will. But they found that there were bony lesions in about 7% of heavily manipulated mice. And they believe that was probably, again, inadvertent discomfort potentially with handling animals and maybe being more traumatic than intended. It was just unexpected finding. And a lot of times, these were not veterinary reported cases. These are just animals that had an issue and then healed independent of veterinary intervention because it wasn't recognized. There were some fractures and dislocations that were noted that, again, we just found were postmortem changes. They were confounding factors. And I think from a veterinary standpoint, as we discussed this within our vet group in my own department, it was realizing that there probably had been trauma and injury that was unrecognized from handling, in addition to pain, which we all know could influence experimental outcomes, and then the knowledge that potentially those injuries or pain was untreated, which goes against our best veterinary practice. So what really came from that discussion, and I wanted to highlight it in this paper so people were aware how important training is for all animal handlers to make sure we have refined and gentle techniques in order to just improve, and particularly for rodents. But I think anyone who's a bit fearful can be more aggressive or tentative than they intend to be. So I feel like training is probably the number one thing we can focus on to make sure that our animal handlers have more comfort working with the animals themselves as patients. And it actually has been cited as the primary area for welfare improvements in a recent survey of research animal veterinarians too, Jenny.
Wow. That's great. Thank you for that summary. Yeah. Like you said, hard to read, but also really important. And it just goes along with that awareness, right? I think it's just shedding more light on some of the things that we do day to day and how that could impact research. So I'm glad you took the time to summarize that for us.
And now it's time for a short break.
Ready to boost your team's skills in lab animal science? Discover the AALAS Learning Library. Train all your staff in one place, from investigators to managers, and earn AALAS, CEUs, and RACE CEs with discounts for groups. Sign up for free to access select courses instantly and explore valuable resources to enhance your team's expertise. Elevate your learning with AALAS Learning Library. AALAS is now on all your favorite social platforms. If you want to be in the know around upcoming deadlines, highlights from this month's journals, or even just fun holidays, follow us on Facebook, LinkedIn, and Instagram today.
So now for our listeners, I don't know about you, but this is the first time I have read in a scholarly research paper a really great analogy on the description of a recipe making chocolate chip cookies and relating that to scientific research protocols. I'm cool with it. I'm definitely down for that. And seriously, I really love this analogy. It made a whole lot of sense to me. And I thought, wow, I had never thought about it that way. But it absolutely did. And I think what you really did a great job of is bringing up the true elephant here, which is generalization versus reproducibility. And just sort of talking about that, I would love to hear your thoughts on that.
Absolutely. So honestly, I have used this recipe analogy with colleagues for years. And as I was thinking about this, I'm like, I'm finally going to try to get this in print because I think it's, yeah. And there was maybe even a little pushback from the editorial staff, but I persevered and I'm glad of it only because, especially in a visual, like when I've given talks on this too, Jenny, I'm able to put a picture of like 4 different cookies. If you just look across like recipe sites online, for example, they always put this like gorgeous photograph of the baked good, for what you're aiming toward. And they never look the same. Like they can be darker in color, or there's more chocolate chips or not, or they're larger or fluffier, right? So the point I was making was that generalizing that if I put up 4 pictures of a round object with dark spots in it, people can say, oh, that's a chocolate chip cookie. Like we all understand that, but it's not the perfect exact same cookie. And I have to share again, like really where this came from, just to be honest, is I was a kid that was in 4-H. Like I imagine many of our listeners probably were. I was grew up in the Midwest.
Yes.
And it was a summer activity. And I mean, I could probably still recite the Heart's Hand health thing if I needed to, but I won't today. But one of the activities that I tried was baking, and you had to do a perfect plate of snickerdoodles. So I'm really hoping other people out there understand that this was very stressful because you weren't allowed to use like a cookie cutter. You had to try to get the dough perfect between each of your cookies. I was terrible at it. I was just awful. Like, I mean, the cookies tasted good, but they looked terrible. I never got more than a red ribbon. Like, let's just put it that way. So when I was really thinking about this analogy, it was like, yeah, I knew those were snickerdoodle cookies and anyone else would have said they were. And you could look across tents, like big summer fairground tents of plates of cookies, but they were all a little different because the people that baked them were different. And that really is kind of what this whole analogy is. And then to draw it even more specifically into science, a recipe is in essence what a protocol is, or a standard operating procedure. It's a listing of steps of how to do a particular action, if you will. And no matter who we put in charge of that recipe or that protocol or that procedure, there are going to be nuances in how someone may enact them, whether they don't follow the exact timeline of every 4 hours exactly they've gone in to give a particular treatment. There might be some range that they had to go in at 3 hours or 5 hours, even though the protocol said 4 hours. Did that get documented? Did that get written down? Did someone oversleep and not come in when they were supposed to do a particular action on the animal? So these are just human nature. These things happen all the time. So I was just trying to make, all in all, the assessment, the analogy, if you will, in kind of a way that was able to be understood by someone who had no idea about science, when we put people into the role, we're generalizing what's going to come out of it. We may not be able to exactly replicate or reproduce what someone else did.
It's a brilliant analogy. I love it. I'm hungry. I'm hangry now thinking about it. And I also am not going to get that visual out of my head of, little 4-H Claire making her cookies, sweating out.
It was ugly. I'll just tell you right now. But now I'm just getting hungry because I can see all the different chocolate.
Well, I'm glad you persevered for sure. I don't know if you invite me over tomorrow and make cookies, I'm not sure I'll eat them, but
I'll be better than when I was in 4th grade. I
appreciate the sentiment for sure. All right. So I guess my next question relates to the question before last, kind of back to that. We were talking about animal handling and some of the studies that you had talked about. And I really appreciated your comments in the paper around human error and just about how error could relate to inconsistency and not necessarily disregard for animals. And I thought that was such an important point that you made, and I would love for you to kind of maybe elaborate that if you could.
Sure. Well, I think probably many people have heard that quote, to err is human, right? So it's kind of a saying like to go off a path is human nature. And I was looking into this, and there's been a lot of discussion around this particular quote as it applies to human patients and inconsistencies in care that can be delivered to human patients, which completely to me translate to how we work with our animal patients as well. And so even if we had all of the same methods and equipment and materials, getting back to this idea of individual human behaviors, when we put people into the mix, there's still going to be an imprecise replication of effort. And so that could be like in the research setting with how people make up their different substrates or how they prepare to do their work. And it could also be the outcomes as well. And so as you mentioned, like I use the term error, ERR, error, in the context of the overview for inconsistency, because if you take the quote a little bit farther, what we don't tend to get to hear, and from the truncated to err is human, if you take that a little bit farther, it goes on to say that it's the clarification of, quote, personality, gender, motivation, and other constitutional factors that give rise to variation, which in turn begets uncertainty and unpredictability, end quote. It's a pretty powerful quote, ooh, Jenny. Like
I love that. Yeah.
It's really good. And I think it's important in this context to have that whole piece, because we're talking about just the uncertainty and unpredictability. So where I kind of took that to the next step, and this was from working again in academic research with many amazing and wonderful individuals that have done animal care, all the way up to the veterinarians who help to provide the treatments, to our research teams. I've done research myself. I'm sat on research groups as well. But we have to think about the human factors, and that was what I was getting to in this. So fatigue, stress, sometimes not having all the knowledge, that can be related to training, as we discussed before. There can also be kind of environmental and organizational factors like distractions, poor teamwork, maybe a management concern. And these distractions, which is really where I started to drill down in one of the sections of the paper, are those that are going on, I think, more than ever before in our society, in our sort of post-COVID world. This was something that I really wanted to try to highlight because distractions can lead to a loss of detailed focus on the task at hand. And that's where inconsistencies can happen. I don't want to call them mistakes, but just, if someone is distracted by a conversation, let's say in an animal room, or they might have headphones on or cell phones, and they're getting a call from somebody, or they're getting texts that they're supposed to be responding to. Those sorts of things can just be distractions that take away from what needs to be done at the time when working with that animal. So as positive and exciting as it can be to have a career working with animals, we also have to recognize that distractions, fatigue, and what we call compassion fatigue and compassion resilience in the veterinary world have just been exacerbated in the last few years, particularly with the COVID-19 pandemic. And in many cases, I think this is because, I don't call it out specifically, but I think so many of the listeners will know, our animal care personnel and veterinary teams were essential and had to still come to work and take care of the animals all through the pandemic, when many people had the opportunity to stay at home. So many of the people in the AALAS community did not have that option because of their commitment and care for the animals. So we're still seeing a lot of fallout, I think, from that. And compassion fatigue can be an occupational hazard. It's actually been identified as that and called as such. And for those who maybe don't know the term, I'll just say really quickly, Jenny, that to define it for folks, it's when empathy, compassion, and caring for others at the core of your work practice wane over time with consequences that can include difficulty sleeping, chronic ailments, depression, and burnout. So we're doing everything we can to recognize this component for animal personnel, again, people that work with animals in some capacity, and then try to combat it with resilient efforts as well, which is making sure people have social support, time to take a break from being in the animal rooms themselves, making sure that there's self-care support, and just trying to mitigate behaviors that might lead to these inconsistencies during times of interacting with animals.
Thank you for summarizing that, especially adding in the compassion fatigue, because I think it's like you, I think it's just so important, and it's something that we really still need to keep talking about. And I really appreciate that in the context of the elephant in the room, it's something that we still have to talk about. So we're getting kind of near the end of our time, but I did want to ask you about the ARRIVE guidelines. As an editorial team member, we talk about these a lot and how best to incorporate them into the AALAS journals. And I would just love to maybe hear your take from what you learned on the paper and what specific guidelines you feel like this paper would sort of help.
Absolutely. And thank you so much for asking about them. I am also a nerd and geek in how much I love the ARRIVE guidelines. So in defining the alphabet soup, so the word ARRIVE in this context is all capitalized, ARRIVE. And for the listeners, that is shorthand for Animal Research Reporting of In Vivo Experiments. So that's how the ARRIVE guidelines came around. They were a huge topic for our NIH working group. I just want to also give a shout out for that too. And I really dug into them even more so because of being a member of that committee for the NIH. But there is a lot of work, a lot of incredibly thoughtful work that's gone into what these ARRIVE guidelines are. And anyone can just Google them, Jenny, and find all the details. These are completely publicly available as well as in print. But the ARRIVE guidelines, just to give like a high level, what are they for? They are a checklist of information to include in publications describing animal research. And the whole goal of that, when they were first established, which I think was in around 2010, and then the ARRIVE 2.0 guidelines came out around 2020, 10 years later. But the hope was that studies would report enough detail to add to the knowledge base and allow in scientific publications, people reading them to then repeat the work. They have enough information, basically have all the steps in that recipe, if you will.
Right. Good. Yes.
Yeah. So the way that the ARRIVE guidelines, the 2.0 guidelines have been digestible, I think is the best way. There's an essential 10, which are 10 big, broad umbrella buckets of where people should think about describing detail, from like study design to sample size to randomization, things like that. And within those essential 10, if you asked me like what would be an area to focus on related to personnel and the personnel influences, probably would be around experimental animals and experimental procedures, just to describe what you're doing with them, what pharmacologic interventions have been done, has any surgery been done, what type of equipment did you use, euthanasia methods as well, even to that point. So that's one of the big bucket parts of the ARRIVE guidelines. The second piece to the essential 10, the complement, if you will, is the recommended set. And when we looked in the recommended set, I would very much advocate that we need to look at a couple of different areas, which again drill into very specific details. The number 15 outlined in the recommended set is housing and husbandry. And this is everything like we just talked about earlier on: the cage, the food and water, the bed, bedding and nesting material, biosecurity, lighting, sex of the experimenter. We didn't get into that too much, and we probably don't have time, but we'll leave that as a teaser that I highlight in the paper. Their animals react differently to whether there is a male or female scent in the room, interestingly. So those things are really important. And I will say, and I'd love to hear your thoughts too, Jenny, from your time in academia too, I rarely get asked by research staff to give details about what was the feed, what was the bedding, what enrichment, who were the vendors, et cetera. Those things just don't make it into publications. And I strongly believe should. And I think AALAS journals do a better job than most of asking that of authors.
Yeah, I definitely appreciate, kind of that summary there, and agreed wholeheartedly. It's been a long time since someone asked about the type of feed, water, all the things, which is always a bit concerning, right? When you're making pretty.
No, the thing is, like, we have it. Like, we have all that information, and that was something else that the working group was stressing. And I was actually sharing this as a veterinarian in research to the scientists on the committee, that for like AALAC oversight and documentation of daily room sheets, we have all this information. Should the researchers want to come back and ask, like, maybe what happened on a particular day, like, oh, the lights got left on for an extra 3 hours. We weren't aware of it because it was after all of our team had gone home for the day. There's lots of ways that info.
And I just keep going back to that quote on awareness. I found in my experience, it was, I would tend to have conversations about very small, maybe picayune-ish things that maybe didn't impact the extrinsic factors, and the rest would just be glossed over, right? And I think, we're kind of saying the same thing, but I found like I would get a little frustrated when the questions would just be about one certain thing that probably wasn't that big of a variable, and the rest would just go on, unaware. We were unaware, or didn't have a great partnership awareness of saying, like, hey, this could also be a factor. And you're making large programmatic decisions about research and resources for research, and I would always tend to be a little flummoxed sometimes when the conversations, and I think it just was, my job was to make folks more aware. And I think this paper will go a long way in giving something for our veterinary groups, our laboratory animal programs, to be able to give to their PIs and say, hey, here's just something for you to sort of talk about. I'd love to have a conversation with you about some of these things. And I'd love for you to know that we have all this data. And, and
even with the data, though, I mean, it's still getting to something that we sort of dance around all those hard facts, the objective, things that we're pulling kind of from these building systems, right, on temperature and humidity and all these other things, all the check boxes that we go through day to day. But we are never going to be able to control for all those things across facilities, because the housing, the husbandry, the personnel, hiring of new researchers, hiring of new animal care people, the diversity of buildings, HVAC systems, seasonal changes, like all regional specific vendors, like it all differs, right, Jenny? So it's
absolutely
it's that awareness piece. I love that we keep coming back to that during our conversation today, because it's sort of like it's just not been talked a lot about in our world. But like when I looked online for sort of observer influences on animals, there were thousands of publications in lots of different disciplines outside of research animal medicine that document how behavior and animal reactions change just by the fact that there are people around them. So think about going to the zoo, for example, or think about coming home from work and how excited your dog is. Your dog was not like jumping up and down behind the door all day long. It's only because you're coming home, like feeding your fish. They all come to the top of the water, right? Like think about all the different ways that animals are influenced by people being around. And I am just excited to have this opportunity from AALAS, from JAALAS specifically, to point out this is a huge influence for what's happening in our research worlds with our animal patients as well.
Yeah, I couldn't agree more. And we could talk about this all day long, and I love it. I'm sure our listeners would love to hear more as well. But unfortunately, we do have to wrap it up. I always ask one sort of final question from my guests, and in this case, I think I want to ask you this. If you were approached by a new researcher, so a newbie, somebody new, doesn't really understand our world, and they're going to start planning a research study, what would you tell them as far as the animal piece?
I think it's a great question, and hard to drill down to maybe just one thing, as you can imagine.
Right.
But I will say, I got asked a question once by a researcher, like, what would be the number one thing that you believe would change and improve animal welfare? When I was looking at coming back to the University of Pennsylvania, that was an interview question I got. And my immediate gut response, which even I would say holds up more so now that I've done this deep dive into personnel practices, is on training and making sure that our research staff, our animal handlers, are comfortable working with the species that's going to be part of their research project. Because that level of comfort, I think, leads to more gentle handling, leads to positive interactions with the patient, the animal patients, leads to more comfort being in the housing rooms, holding the animals and manipulating them, training people how to do a blood draw, how to use positive reinforcement to have non-human primates present for their own, treatments, et cetera. I think training around animal care is probably the number one thing. And for any new researcher coming in, or even established researchers, I think reaching out to the training groups at the institutions you may be at, and just checking in, are there any new things you're trying? NC3Rs has really been pushing support for rat handling. They're calling it rat tickling, and many in our industry will be aware of that term, but you can get a certificate in that now. There's just a lot of different ways to amp up the training effects. And not making it be a regulatory burden, which I always hear as a negative, but actually investing the time in your people so that they're going to be successful for the research animals. And also it improves animal outcomes and data as well, I believe.
Oh, I couldn't agree with you more. And I so appreciate you covering that. It is critical. And I so appreciate you summarizing in that way. And yeah, I couldn't agree more. So unfortunately, we are at the end of our time. I hate to end because, like I said, I think we could talk all day about this and then talk about everything else after that. So thank you for taking the time out of your busy schedule to be with us. I know our audience learned a lot. I did as well. So for everyone listening, we invite you to subscribe, leave us your comments and questions in the section below. Additionally, we invite you to consider submitting your current or future research to the AALAS scientific journals. Those are JAALAS or CompMed. Please follow us on our socials, or you can always email us, info@aalas.org. We really do want your feedback, and we want to know what you think about this episode. And finally, thanks again for listening to the Last Word in AALAS podcast. Be on the lookout for information on future podcast episodes dropping on the 1st of every month.